So this is our video explaining in vitro amplification. Amplification is the copying of DNA fragments many a times to make millions of copies of a fragment of DNA. In vitro means it occurs outside of a living organism's body, and this is done by the polymerase chain reaction. Our video will explain how the polymerase chain reaction occurs.
This test tube illustrates a reaction mixture which contains a DNA sample, 3 nucleotides, primers, and DNA polymerase. Primers are short pieces of DNA that are complementary to the bases at the start of the DNA fragment that you want.
The DNA mixture is heated to 95 degrees to break the hydrogen bonds between the two strands of DNA.
The reaction mixture is cooled to a temperature between 50 and 65 degrees Celsius. In our diagram it's at 55 degrees Celsius. This is done so that DNA primers can bind to the strands.
The reaction mixture is then heated to 72 degrees, which is shown here, so that DNA polymerase, which is an enzyme, can work. The DNA polymerase lines up 3 DNA nucleotides alongside each template strand. DNA polymerase is illustrated here with a white dot. Specific base pairing means that new complementary strands are formed.
Due to this, 2 new copies of the fragment of DNA are formed, and 1 cycle of PCR has said to be complete.
The cycle starts again with the mixture being heated to 95 degrees C, and this time all 4 strands, 2 original and 2 new, are used as templates.
For each PCR cycle, the amount of DNA fragments double each time. For each PCR cycle, the amount of DNA fragments double each time.